All right, guys, so we are going to be looking at the immune system in this section here. This session is going to be on immunity and the various questions they can ask on it. All right, so one of the first questions I saw was a question on monoclonal antibodies. So I decided to just remind you what monoclonal antibodies are and how they are produced. So normally, guys, an antibody is produced by a B cell, and the antibody is supposed to bind to a specific antigen. So one antibody that binds one specific antigen is called a monoclonal antibody. So this is the trouble now. So you can't actually have, when a B cell actually has met an antigen, the B cell will, well, multiple B cells are going to try to see which one of them can best destroy the antigen. So you find multiple clones of B cells trying to destroy the antigen, and then one antibody might actually fit the best with that antigen. So that specific clone will be selected, and B cells cannot secrete antibodies. So even though B cells have antibodies on their surfaces, they cannot secrete those antibodies. They have to first be converted into a plasma cell for the plasma cell to be able to secrete the antibodies. And unfortunately, plasma cells are not able to copy themselves. So it's very difficult for you to get copies of a specific antibody outside of the body of the organism. So this method of producing monoclonal antibodies was designed whereby we would have gotten, well, first we would have injected a mouse with the antigen that we want to create antibodies against. And we would eventually take cells from the spleen of the mouse, because we are hoping that with time, the mouse would have created antibodies against the antigen that we injected. We also at the same time get a culture of cancer cells, myeloma cells. Myeloma cells are cancerous B cells. So what we do now, they love to divide. So what we do is we fuse the spleen cells, which are able to produce which have the antibodies on their surface, with the cancer cells, and we're hoping that they will create this new type of cell known as a hybridoma. And the hybrid cells, we will be able to make copies of them, and they will produce antibodies for us. Sometimes they can even inject back the hybrid cells into the mouse for the mouse to create antibodies. But this is how we can create a large amount of one specific type of antibody, right? A monoclonal antibody. So what are the purposes of these antibodies? They can have diagnostic applications. So if you have an antibody against a specific protein, the antibody can help to detect that protein of interest. You can use it to detect areas where there are cardiovascular diseases. You can use it to detect when you have clots. You can use it to identify primary tumors and secondary tumors. You can use it as immunosuppressive therapy, and even as pregnancy testing kits, which we'll discuss shortly. It can be used therapeutically to help to treat certain cancers, because the antibody can go and target the cancer cells. So if you can attach the medication to the antibody that is made against the specific cancer cell, then you could deliver the medication to the cancer cell only without damaging normal cells. All right, so the main therapeutic one I'll ask you to remember is how it helps to target cancer cells. So there was a question in the 2021 paper on immunology, the applications of biology, where it's asking you about monoclonal antibodies used in early testing kits for pregnancy. And it shows a strip from a home pregnancy kit, and there are 3 areas in each strip which where the reaction occurs. So you have the person would have normally had passed urine, and the urine would have been applied at number 1, well, down here. So there are 3 bands in the pregnancy kit. So we have 1 band, that has a monoclonal antibodies that were made in the mouse, and this would be able to detect a specific hormone in the woman's urine if she's pregnant. So the antibodies would be able to detect beta HCG. So if she has beta HCG in her urine, then the antibodies would bind to it. And the antibodies that are bounded to the beta HCG would continue on the strip. Now, this second part has many different types of antibodies and dyes, and number 3 contains anti-most antibodies and dyes. All right, so this test, the second strip actually is supposed to be the control strip. To assess the functioning or the proper functioning of the strip. So for this one here now, you just want to know if the antibodies made its way down here. Because remember, you have monoclonal antibodies from the first part of the strip. So you are supposed to see if any of the monoclonal antibodies made its way down here, so you know that the strip is working. But the part number 2 now is supposed to be able to bind to monoclonal antibodies that have beta HCG bound to it. All right. So I'm going to come back to this pregnancy test here because I wanted to show you another version of it. So there was a question asking you to define a monoclonal antibody, the 2 marks. So monoclonal antibodies are antibodies that are identical because they were produced by one type of immune cell by a single parent. All right, so how does this whole thing work now in pregnancy test kits? So bear in mind that we're going to be answering this past paper question, but I kind of wanted you to visualize what was happening first. So if the woman is pregnant, she produces HCG molecules, all right? Then, remember we have these antibodies that are they can bind to HCG. So these are in the pregnancy test kit. So they have a site for the HCG to be bound, and they have another site for enzyme activity. All right, so there's an enzyme attached to the antibody against beta HCG. So if she has HCG in her urine, then it's going to bind to the antibody that can bind to HCG. So then now, these are going to travel up the pregnancy kit. So HCG molecules are carried up in test urine. HCG molecules bind to mobile antibodies to form HCG antibody complex. So these antibodies are mobile. So the mobile antibody complex will bind to, when they get to this part, they're going to bind to immobilized antibodies and enzyme catalyzed reactions that results in colored compounds to form a colored line indicating pregnancy. So at this line here, we have these antibodies, which are called immobilized antibodies. This antibody binds to beta HCG that is bound to the first antibody. So it can't just bind the beta HCG by itself like that. It binds beta HCG that has already been bound to the other antibody. So it's binding the antibody complex. Now, remember that there was an enzyme attached to this antibody here. So what you do is give the substrate for the enzyme, which is an inactivated dye. And when the enzyme acts on it, it converts it into the colored dye. So if you see this strip coming up on the pregnancy test, it simply means the woman is pregnant because it means she had beta HCG bounded to these mobile antibodies that came to get bounded to these immobilized antibodies, and the color reaction was successful. Now, the other strip is a test strip. So there is always more mobile antibodies present than HCG molecules. So you should always have some mobile antibodies left over that would not have any HCG to bind to. So the leftover mobile antibodies, those are what you are testing for in the last strip. So the excess mobile antibodies will bind to different immobilized antibodies And the enzymes catalyze a reaction that results in a colored compound to form a colored line, because the antibody binds whether it has formed a complex with HCG or not. This line will appear even if the woman is not pregnant. So here we have another set of immobilized antibodies that bind to these mobile antibodies from down the bottom here. So it's just checking to see that the mobile antibodies from the first part have actually traveled down the strip. So whether or not it has HCG bonded or not, it will still bind to these antibodies down here. Notice it's binding to the bottom part of the antibody. So it doesn't matter if there is HCG attached or not. And remember that there is an enzyme attached to this one, so you can add the substrate and it should give you a color. So it simply means that a woman is pregnant if she has both lines. If there is just one line and this one doesn't give a color, then it means she's not pregnant and the strip is working well. If this first line shows up and nothing shows up in the second line, then you can't consider that a valid pregnancy test, right? You must see both lines for you to say, yes, the strip is working. Yes, she is pregnant, all right? So now this is the question that arises from what they were talking about for the pregnancy test now. So figure 6 is a representation of the reactions that occurs in the home pregnancy test strip. Well, you have reaction A is a dye, wait, the circle is the dye. Then this is HCG, reaction B. And then this is the antimose antibody, the pale ones. These are different antibodies. And then you have monoclonal antibody with the enzyme attached. So now we are supposed to identify the areas labeled 1 to 3 and figure 5 on page 16, where the reactions A, B, and C would occur. So reaction A now is just showing us the antimose antibody binding to, no, the monoclonal antibody binding to the antimose antibody. So this is reaction A, right? So they're asking us where, so this was the one I kind of showed you as the illustration. So they're asking us where on this strip is reaction A taking place? Is it at number 3, 2, or 1? I want you to look back at the example. Then look back at the reaction A and tell me where should I say that reaction A is taking place? Yes, guys, I'm waiting to hear from you.
Miss, can you scroll back up for a second?
So look back at reaction A. So reaction A is basically, I'm going to help you with this one. This one is just basically what you would see at the test strip. You're just checking that the mobile antibodies made its way down the strip. That's all it's doing. Look at reaction A and see if it doesn't match up with what you're seeing here. So these are immobilized antibodies binding to mobile antibodies. That's it. Just checking to see that the strip is working. So it's very similar to what we're seeing here. So reaction A would be occurring at number 3. All right, so we've done that. So let's go back down. Look at B now, where the mobile antibody is binding to the beta HCG molecules. So which strip would that reaction B will be found at which strip now? That would be at reaction number 1. Yeah. So that is just the first test, just to see if the lady has beta HCG in her urine, then you should just have the mobile antibodies binding to it. So this would be reaction B. And then clearly the other one would be at test number 2. So this is where the person, the first, she has beta HC bonded to the antibodies. And then you have but antibodies bonded to the ones which have beta HCG. So that's what would tell you that she's pregnant, which is what was at strip part 2, at the part 2 of the strip. So that would be reaction C right here. All right. Identify the area. So we said reaction A would have been at number 3. B would have been at number 1, and C would have been at number 2. So then now we are to outline the processes which would occur in each labeled area in figure 5 if the pregnancy test were positive. So number 1 is where we have the mobile antibodies with enzyme attached. Bind to beta HCG molecules in the urine to form a HCG antibody complex. Okay. The second thing is number 2 now is where we would have had the immobilized antibody binds to the beta HCG antibody complex. The substrate to the enzyme is then added for a colored strip to form. And in number 3 now, mobilized antibodies bind to mobile antibodies. And then the substrate for the enzyme is substrate for enzyme. So the substrate for the enzyme is added, which causes a colored strip. Okay. So next we have this question on vaccination. So 2019 question 3B says vaccination can be used to control the spread of mosquito-borne diseases. With the aid of a labeled graph showing blood antibody concentrations over time after vaccination, explain 3 ways in which vaccinations work to reduce the spread of diseases. All right, so this one, I'll just discuss the answer. All right, so vaccination is a type of it's artificial active immunity. So essentially, we are given the pathogen in a weakened form, or we're giving a part of the pathogen, exposing it to the person's immune system. Then we're hoping the immune system would have identified the pathogen, would have determined the clone of B cells that are suitable to create the right antibodies, would have made multiple copies of those B cells. And then you would have memory cells as well, because every time the B cells divide, they normally make memory cells as well. So the memory cells would be there. So you're expecting that the first exposure to the pathogen, you're going to have a very slow response. That's a primary response, because it's going to take time for you to figure out what clone of antibodies you need in order to give you a successful binding to your antigen. And then once you figure out the right clone, then you would make memory cells. So it will take a few weeks for you to destroy the pathogen in the primary response, but because you would have the memory cells, then if the pathogen comes back for the second exposure, then your memory cells will rapidly become plasma cells, and you can produce large concentrations of the antibody in a very short period of time. That's a secondary response. So you get marks for correctly labeling your axes. Make sure the graph has the right shape. So you should make sure you show that the secondary response is larger and quicker than the primary response, all right? The time should be more or less, it should be like in days or even weeks, days to weeks, because to be honest, the first one would take weeks, but the secondary response would take days. So I guess that's why they didn't specify the unit of the time. So after you have drawn the graph, you are to explain now how vaccination helps to control disease. So vaccines, where you give the antigen to trigger a primary immune response in the body, you'll have a relatively slow increase in antibodies in the blood that can destroy the pathogen and prevent disease from spreading. The antigens also trigger the production of memory cells, both B and T memory cells, which should remain in circulation to provide long-term immunity and to prevent the spread of the disease. The secondary immune response is triggered if the body encounters the same antigen at a later time. That's what is called immunity. So both your B cells and your helper T cells had made memory cells from the first encounter. So these cells will promote larger and faster increases in your antibodies to prevent the disease from spreading. The T memory cells will activate T killer cells also to destroy infected body cells to prevent the disease from spreading. So there will be fast destruction of pathogens by the antibodies and the cytotoxic T cells to prevent the spread of disease, and so the immunized person cannot spread disease to others. There is also the whole concept of herd immunity, where vaccination helps decrease the amount of possible hosts for the pathogen. So there is a low chance of disease occurrence if a high proportion, up to 80 to 85% of your population is vaccinated. The larger the number of vaccinated people in a population, the lower the chance that anyone will get the disease, even though some members are not yet vaccinated. There is also the concept of ring vaccination, which is a strategy to inhibit the spread of a disease by vaccinating only those who are most likely to get infected. So it controls an outbreak by vaccinating and monitoring a ring of people around each infected individual. It forms a buffer of immune individuals around the infected individual to prevent the spread of disease. So they're basically saying you can create immunity in individuals that helps to decrease the spread. You can also get herd immunity in a population that decreases spread, and you can also use the ring vaccination technique where you vaccinate individuals surrounding the person who has been infected to create a buffer zone. All right, so then this slide now was to remind you of the types of immunity. Oh, well, I really should have done. All right, so this is talking about acquired immunity that you acquire over a lifetime. So you're born with innate immunity. So that's what you're born with, but over time you need to have acquired immunity. So acquired immunity, which develops in your lifetime, could be considered to be active or passive. So active immunity is one that develops in response to an infection or vaccination. So that means that you have been exposed to the pathogen. Now, passive immunity develops after you receive antibodies from someone or somewhere else, meaning that you are not exposed to a pathogen. All right, so then now, in active immunity now, which is, it could be natural, where the antibodies develop naturally in response to an infection, or artificial, where you developed antibodies as because you were vaccinated. But the good thing about this is that you have memory cells. So memory cells are produced. So there is long-term protection. All right, passive immunity now develops after you receive antibodies from someone or somebody, somewhere else. It could be natural that you received it from your mother through breast milk, or artificial that you got the antibodies from a medical facility. But the thing is that no memory cells have been produced. Right, so it only gives temporary immunity. So then you're asked to distinguish between natural active immunity and artificial active immunity. So we just spoke about natural active immunity. So antibodies develop in an individual after they have been exposed, after they got an infection. So that would be the natural active immunity. And then the artificial active immunity, so antibodies, I should say antibodies and memory cells, antibodies and memory cells. So in this one now we have antibodies and memory cells. developed in an individual that was vaccinated. That will be artificial active immunity. All right, next. Vaccination is regarded as a form of active artificial immunity. So distinguish between the terms active immunity and artificial immunity. So active immunity means that there is stimulation of a person's immune system by an antigen or pathogen to make specific antibodies, and it can provide long-term protection against antigens because there is a formation of memory cells. So once you have stimulated the immune system to make memory cells, that means that it is an active process. The term artificial means that the immunity was acquired by deliberate exposure given either by vaccination or antibodies to an antigen or pathogen. So it was not, you naturally were exposed to the pathogen, it was deliberate. So that's what the active mean, and that's what the artificial means. Use examples, explain the role of vaccination in providing immunity to pathogens. We kind of discussed it in the previous question, but it doesn't hurt to discuss it again. So vaccination prevents persons getting infectious diseases. And remember, herd immunity can allow masses or a population to be immune. So you have the individual becoming immune, plus you're also trying to get an entire population to be immune. So vaccination involves introducing a small amount of a pathogen into the body through a needle by injection, or it could be done orally or by aerosol spray. There are different types of vaccines. There are live attenuated vaccines where the living organism has been given, but it's weakened, so it's harmless. For example, measles, rubella, tuberculosis, yellow fever are live attenuated vaccines. You could be given the dead pathogen, for example, in polio, or you could be given toxins that the pathogen produces like tetanus and diphtheria, or you could be given fragments of the pathogen, for example, pertussis and hepatitis. Most vaccines may require more than one dose. For example, tetanus requires a booster needed for most vaccines over time because immunity may not be lifelong. When antigens enter the body, the white blood cells, lymphocytes will respond to it as if they had encountered the live pathogen. So plasma cells will be stimulated to produce antibodies against the specific antigen, and memory cells would also be formed in addition to plasma cells that will remain in the circulation in the body for long-term protection. Usually the person does not become ill or experience only mild symptoms of disease if they encounter the pathogen at a later time. So if persons later become infected with the pathogen, memory cells are able to rapidly produce antibodies. The immune system reacts faster and produces more antibodies. That's a secondary response. So that is how we can speak about the role of vaccination in providing immunity to pathogens. All right. So it's for 6 marks. So they wouldn't necessarily expect all the points. I would definitely say that you definitely would want to include this point here, this point here. I wouldn't necessarily go into the different types of vaccines. I would then talk about this. And then the other points that follow are important. So if you were to give 6 points, I would want to go with these 6 that I have highlighted as mandatory points to get the 6 marks. All right, so we're moving on to a discussion about humoral and cell-mediated immunity. All right. Let's look at this one. What is the difference between humoral and cell-mediated immunity? Humoral immunity is basically executed by B lymphocytes, whereas cell-mediated immunity is by the T lymphocytes. Where do cells develop? The B lymphocytes are produced and they are produced and they mature in the bone marrow. And the cell-mediated immunity, they are produced in the bone marrow and they mature in the thymus gland. So humoral immunity involves the production of antibodies, while cell-mediated immunity does not involve the production of antibodies. So how do we identify the pathogens? So normally the B lymphocytes would identify the pathogens as they are floating around in the blood. But for the cell-mediated immunity, the antigens have to be present on the surface of infected cells or on antigen-presenting cells. So the T lymphocytes cannot just react to an antigen floating around. It has to be on the surface of a cell for it to respond to it. It has to be presented to the T cell, whereas the B lymphocyte can see the pathogen and respond. The T cell has to be presented to the C cell. How are the pathogens killed in cell-mediated immunity? They are killed by the antibodies. In cell-mediated immunity, the pathogens are killed by cytotoxic T cells. So the infected cells, infected body cells are destroyed by the cytotoxic T cells. So really and truly, we mostly use the cell-mediated immune system to destroy intracellular pathogens for example, viruses which come and live inside the cell. Some protozoa too, they love to come and go inside the cell. We also use it to kill cancer cells because these are our own cells, right? But we use the humoral system to destroy extracellular parasites or pathogens. All right, so how do cells divide once they are stimulated? So the cells divide and turn plasma cells or memory cells. And if the cell-mediated immune system is activated, the cells divide into specialized T cells. So they could be T killer cells and T memory cells. Let me just put that in. So T killer cells are all right, so another way I have put a slide here to also tell you more about the differences. So the humoral immune system will attack intact antigens. Once the B cells have encountered an antigen, the B cell always communicates with the helper T cell, always. So the helper T cell is the coordinator of the immune response. So if the B cell has encountered a pathogen, it does tell the helper T cell that it has encountered the pathogen. What the T helper cell will do, it will say, all right, B cell, you are the one who should take care of this pathogen. And what the T cell does is that it releases chemicals that enhances the activity of the B cell. So if the B cell were to go ahead and try to destroy the pathogen by itself without telling the helper T cell, the B cell will not be very effective. So it displays the pathogen, tells the helper T cell, and the helper T cell will in turn support the B cell as it divides. So the B cell will eventually, you'll find the right B cell that has right antibody. And once you have found the right antibody on the right B cell, then you make clones of that B cell. It will form plasma cells, which actually secrete antibodies, and memory cells, which will stay behind and circulate in the bloodstream. So then you have, what if the pathogen is intracellular? So you would have needed to have the antigen displayed to the helper T cell by one of the antigen presenting cells. Maybe it's a macrophage, maybe it's a B cell, something, or the infected cell would have made some receptors or some proteins to indicate to the helper cell that it has become infected. So when the T helper cell gets information from a body cell that it has been infected, the T helper cell will make chemicals to enhance its own self. So it makes copies of itself to make T memory T helper cells. Plus, it's going to stimulate the cytotoxic T cells, right? So the cytotoxic T cells are the ones who actually can bind to infected cells. So for example, now, say for example, we had a cell that had a virus inside of it. Right, so say there was a virus inside of this cell. This is a body cell with a virus. What this cell is going to do is that it's going to put out some things on its surface membrane, showing some parts of the virus. And it's going to present it to the cytotoxic T cell, right? So the cytotoxic T cell would be able to come and bind to whatever receptor is being given off by this infected cell. And it realizes that the cell has become compromised because it has been infected. So when the T cell realizes that the cell has been infected, the T cell is going to communicate with the helper T cell and say, hey, I have detected a body cell that has a virus inside of it. So the T helper cell says, all right, go ahead. Your job is to destroy this infected body cell because this is cell-mediated immunity to deal with an intracellular virus. So the helper T cell says, I'm going to support you, cytotoxic T cell. So the T helper cell releases chemicals, causing the cytotoxic T cell to divide more rapidly. And the T cell also makes memory cells so that it will remember this pathogen. So the cytotoxic T cell makes memory cytotoxic cells and active cytotoxic cells. These active cytotoxic cells will actually go and kill this cell that has a virus so that it doesn't spread. So here we have a table. 2 is an incomplete comparison of humoral and cell-mediated immunity. Complete table 2 by writing the answers in the relevant spaces. So the humoral immunity is done by the B cells. So the cell-mediated immunity we know is done by T cells, right? T lymphocytes. The mode of action is that antibodies circulate in the serum. But mode of action is that the cytotoxic T cells bind to the infected body cell. And make pores in the membrane thus destroying it. It doesn't only make pores in the membrane, it makes pores in the cell membrane and injects. Toxic enzymes, thus destroying the infected cell. All right, the purpose, it says here for the cell-mediated immune system is that it's the primary defense against intracellular pathogens, tumor cells, and for graft rejection. So for the cell-mediated system, we would say that it's a primary defense against extracellular pathogens. And the onset of the cell-mediated that is delayed, but the human humoral system is basically gives a more immediate response. It's not delayed. All right, explain why after a first infection with measles, a person is unlikely to be reinfected with measles. That's because during the first infection with measles, memory cells were formed. These memory cells continue to circulate in the blood or lymph. So if there is contact with the same antigen, for example, measles antigen, the memory cells will divide and more memory cells will be produced. Memory cells differentiate into active cells, plasma cells, and active T cells to protect against reinfection, giving protective immunity. All right, so with that being said, now we're going to look at phagocytosis because they will ask questions on this. You know, if you know the stages of phagocytosis. This would be done by our phagocytes, which can be macrophages, neutrophils, etc. So say, for instance, there is a pathogen. Well, they say food particle here, but it could have been pathogen as well. Pathogen. The, the phagocyte has to recognize it as being foreign, and then it makes the pseudopodia, false feet, to surround the pathogen and engulf it. And it completes, it makes a vacuole and completely surround the pathogen. Then there are lysosomes, which are filled with digestive enzymes in the membrane of the phagocyte. So eventually, the lysosomes are going to fuse with this vacuole here, and it will be called a phagolysosome. So when you have the when the pathogen is in a vacuole that has lysosomes around it, it's called a phagolysosome. And then when the lysosomes inject the digestive enzymes, it will destroy the pathogen, right? So those are the key steps of phagocytosis. So here we have a question, 2014 question 3B on the steps involved in a phagocytic process in the macrophages. So we have 1, 2, 3. All right. So on figure 5, use annotations to describe the steps labeled 1, 2, 3. So what would you give as the annotation for step 1? All right. So the first step would just be to engulf the bacteria. So pseudopodia are formed around the pathogen. Then number 2, what's number 2? All right, number 2, let me see here. The phagosome is fused with the lysosome. Well, first we have to make a vacuole. All right, so just by surrounding the pathogen completely in a vacuole, so That's when we actually created a phagosome. So a vacuum, all right, I should say the pathogen is completely surrounded by, the pathogen is now then completely surrounded by a vacuum. I should say by a membrane, and it's contained in a vacuum. And then when you get to the phagolysosome now, that's when the lysosome fuse with the phagosome. And then it will give it will and release its digestive enzymes and release its digestive enzyme. And then the pathogen is digested, but you do save back a portion of the pathogen so you can present it to the T helper cell. So how might a macrophage act as an antigen presenting cell? Well, so you want to say after the pathogen has been digested in the phagolysosome, proteins or antigens from the pathogen are preserved. So intracellular receptors will bind to the pathogen or bind to the antigen. And then now the antigen receptor complex is inserted into the cell membrane and presented to the helper T cells. All right, so that's how the macrophages are able to work as antigen presenting cells. So they try to destroy the pathogen, but they keep back remnants of it to show to the image, show to the T helper cells. All right, so this one now I kind of include this to show you how B cells and T cells are formed and how they are mature. So our bone marrow cells, well, the bone marrow, sorry, creates the original stem cell. It's called a hematopoietic stem cell. It is able to form our original lymphocyte or lymphoid progenitor cells. Some of them will go off to form immature B cells and some will go off to form immature T cells. The immature B cells will actually develop in the bone marrow, and after they have become mature, they will then go to the spleen and other areas. But immature T cells go to the thymus. So I really should have, so immature T cells. So immature T cells will go to the thymus. The thymus is where T cells are going to be educated how to recognize their own self antigens versus foreign antigens. And then by the time they leave the thymus, they will be mature T cells, okay? So the key thing is that the B cells are formed and mature in the bone marrow, whereas T cells are formed, but they mature in the thymus. So when they say describe the origin and the maturation of T cells, we say they originate in the bone marrow. From hematopoietic, well, or from lymphoid progenitor cells, precursor cells. Then immature T cells. Go to the thymus for maturation. And then the mature T lymphocytes are released from the thymus for circulation. Yep, so that's how the T lymphocytes would be formed and mature. The next question says, state 3 types of T cells and outline their functions. So we have T helper cells. Their job is to stimulate B cells to make antibodies and stimulate helper cells to kill, to be more effective. So the T helper cells enhance the function of the other immune cells, right? Then you have cytotoxic killer cells, which directly kill cells that have already been infected by a foreign invader. And you have regulatory T cells. They suppress the immune response when it's no longer needed. So after you have destroyed the pathogen, you need a T cell to tell you to stop. With reference to HIV, explain why T cells are sometimes unable to effectively eliminate foreign organisms. I'd like to hear what you think about this specific question here. So HIV is a virus. Why can't our immune system eliminate HIV completely when once it has infected an individual? I want to hear your thoughts, guys. All right, guys. So this is the problem. Let me see what you wrote in the chat. So you have seen, because the virus inserts its DNA into the person's DNA, good, that's good. It keeps changing, so the antibody can't destroy it. All right, not exactly. I wanted you to say that this virus infects the T helper cells. Remember, this is the cell that is supposed to coordinate the immune response, you know, and it infects the T helper cells. Well, part of what you said about incorporating the DNA is good. So the T helper cell is infected, and the virus integrates its DNA into the host DNA. So that's going to be a challenge because your T helper cell has become compromised, plus the virus has inserted its DNA, right? So it is difficult to detect at the infected cell because it's not like the virus is actually doing anything a lot of the times. It's just dormant. So sometimes it escapes the immune system. It's not detected. All righty. So we're going on. So we have another question is going to come up on the complement system. So I kind of put this slide here to kind of remind you about the complement system. So it's a group of proteins, right? A group of plasma proteins that can become activated to form, well, it can do many things. It can become activated to assist the humoral system. So we kind of have so many lines of defense to kind of destroy pathogens. So you have some proteins in your blood, right? Now, if something triggers off the proteins, there's a bunch of proteins. So one protein activates the next one, that activates the next one, that activates the next one. But at the end of the day, the proteins can come together and form a membrane attack complex. So it can destroy a cell by inserting this complex in the membrane. So the cell gets destroyed. Or the complement can actually help antibodies to destroy the antigen more effectively. So the complement system actually helps the humoral system in fighting against pathogens. It's actually a part of the innate immune system as well as the humoral system, right? So here's a question asking now about the complement system. So explain the term complement and outline its role in the body's response to invasion by foreign organisms. So the complement is a group of short-lived soluble proteins always present in your blood plasma. They are activated by contact with antibody bound to antigen or by a foreign surface. Activation of a complement triggers a cascade of reactions, which results in the production of active protein. Its role is that it can do opsonization of bacteria, meaning it coats the bacteria, making it more easy for the phagocytes to destroy. It also can attract macrophages and other white cells to the site of infection, so it can basically enhance the immune response. And remember, I showed you the membrane attack complex. They can sometimes directly destroy a cell by forming the complex in the membrane of the cell, destroying it. All right. So we're pressing on, almost done with this. So the immune system can be broken down into innate immunity and adaptive immunity. So the innate immunity is really what you're born with. We have our first line of defense, or I should say the innate immune system is non-specific. So it's not really trying to identify the pathogen. As it says there, it's non-specific. There is no attempt So the first line of defense is your skin. So the first thing is to make sure you have a secure barrier and your mucous membranes as well. And you can secrete substances from the skin and mucous membranes to try to not allow the pathogen to enter the body. But God forbid the pathogen enters the body. The second line of defense would be to use your phagocytes to see if you can phagocytose it. And you also have your complement proteins now. That's what they mean by the antimicrobial protein. That's the complement system they're talking about. And you will also use an inflammatory response, which we'll talk about the inflammatory response a little bit later. I should have actually included something about it. But inflammatory response is really just your body's way of trying to destroy this pathogen quickly, meaning that you make sure the blood vessels get dilated so more blood can come to the area where the infection is. You also allow the blood vessel walls to become more leaky so that white cells can exit the blood and go to where the pathogen is. So when you have the inflammatory response triggered, you will have warmth, pain, tenderness at the affected area, but it's really just trying to initially contain the pathogen and attract as many white cells as possible to see if you can contain it in that location. And then a fever is when you are actually giving off a systemic response. You are making the entire body know that there is an infection here. When you end up with a fever, it means that it's an inflammatory response, but you have made it become systemic. So the entire body is aware that there is a pathogen inside. Versus if you get stuck on your hand and you end up getting the hand getting swollen and tender and pus forms in your hand or on your hand, but you don't get a fever. It means that you have been successful in containing that infection in that location. When you get a fever, it means that the infection is a bit more serious. It could not have been contained in that one location, and it's now a systemic response is being triggered so that more white cells from all over the body can come and see if they can contain this infection. The third line of defense now is your adaptive immune response now, which includes your humoral and cell-mediated response. Now, your third line of response is to really try to identify the pathogen. Tries to identify the pathogen. So it's not like these, they're not attempting to identify the pathogen, they just want to destroy it. But the third line wants to identify it. So that's where your lymphocytes, your antibodies, and memory cells come into play. All right, so the entry of foreign organisms into the body, for example, when there is a break in the skin, is often followed by redness, warmth, and swelling at the site of entry. Once you hear redness, swelling, warmth, you know that that is an inflammatory response that has been triggered. Discuss why each of these responses is an indication that the body is attempting to eliminate the foreign organisms. All right, so why? When you have redness at the area, it's because mast cells, which are a type of macrophage or type of phagocyte, produces a chemical called histamine, which triggers the arterioles to vasodilate and more blood flow comes to the infected tissue. That's why the area gets red. More blood is flowing to the area. That's redness. Warmth. Because more blood has come to the area, there is more metabolic heat to try to destroy the pathogen. So that's why the area gets red and warm. Why is there swelling? Because the same histamine that was released increases the permeability of the capillaries, so plasma proteins can escape and changes the fluid balance across the capillary walls, leading to swelling in the area. So these are the hallmarks of inflammation: redness, warmth, swelling, and pain. Pain is when you actually start destroying the pathogen, and sometimes some of your own cells get destroyed in the process. All right, so we're almost done with this topic. So this one says, when the human body is invaded by a foreign organism, it responds by identifying and destroying the foreign organism in a manner that involves both a cell-mediated and humoral response. Distinguish between cell-mediated and humoral responses. So cell-mediated, we know that the T cells are used. It involves the entire cell. It involves cytokines. These are special chemicals released by T cells, and they recognize antigen fragments. The humoral response involves the B cells, involves the release of antibodies, so not cytokines, antibodies. It recognizes the entire antigen. The whole cell is not directly involved, just the antibodies destroying the pathogen. But this is a simple way to distinguish between cell-mediated and humoral response. All right, when Kayla was 8 years old, she was infected with measles. 10 years later, while volunteering at an orphanage, she's required to take care of a baby with measles. Explain the immunological process which is responsible for Kayla not becoming ill. So Kayla had gotten a first exposure to measles and had done a first response, primary response. So she actually had B lymphocytes that were formed and she made memory cells. That initial response was slow, but she left back clones of her plasma cells, which were known as memory cells. So clones of plasma cells produce antibodies which bond to the antigens and destroy them. The plasma cells don't live long, but the memory cells ensure that there is a rapid response to future invasions. And so she can mount a secondary response because of the memory cells that are long-lived. And the memory cells will store the information for specific antigens. I should add that the memory cells can quickly become plasma cells. Okay, so the reason why Kayla won't get infected is because she already has memory cells against measles. So if she's taking care of any child at the orphanage who has measles and the measles antigen enters her body, the memory cells will recognize it and be converted into plasma cells and immediately secrete large amounts of antibodies, destroying the measles antigen before it makes her sick. So she is immune. All right, so now to the last question on this topic. Define the immune response and distinguish between cell-mediated and humoral responses. So the immune response is the defense reaction or the mechanism to recognize or detect an invading non-self organism. That's basically what it is. So your ability to detect something that is not yourself or an invading organism and have a defense reaction to it. So cell-mediated versus humoral. Cell-mediated responses are due to T cells, lymphocytes, while humoral are due to B cells. The whole cell or lymphokines involved in cell-mediated response, but antibodies are involved in humoral response. The T cell receptors recognize, bind to fragments of an antigen, whereas the antibodies will recognize and bind to the entire antigen. Discuss the maturation of B and T cells in relation to their specific roles. So B cells So the B cells develop from stem cells in the bone marrow and they remain in the bone marrow and mature there. Or sometimes they can say that they do mature in the spleen as well. But after leaving the bone marrow, B cells circulate in the bloodstream in search of antigens to remove or get rid of. They make antibodies, make memory cells that can, and then the antibodies can destroy the pathogen. T cells, though, even though they're formed in the bone marrow from stem cells, they go to the thymus for maturation. And after leaving the thymus, they enter the bloodstream and or migrate into the spleen or lymph nodes to recognize and eliminate foreign materials, especially those that have infected our cells. Comment on the use of monoclonal antibodies in home pregnancy tests. So we discussed this already. So when one becomes pregnant, newly implanted embryos produce HCG, a hormone which circulates in the blood and is excreted in the urine. Pharmaceutical companies clone the B cells which produce monoclonal antibodies in response to the antigen HCG. Urine from a woman is tested with a pregnancy kit, strip of paper with monoclonal HCG antibodies. If HCG is pregnant, you get a positive test. A line is obtained suggesting the woman is pregnant. And remember, there should be a control line as well. So it enables cheap, inexpensive testing. All right. And finally, explain the difference between active artificial and passive artificial immunity. So active artificial immunity is vaccination, where antigens from weakened, attenuated, killed vaccine, weakened, attenuated, or killed organisms are administered. The immune response is stimulated to produce antibodies. Long-term protection is achieved. Long-term production is achieved. Memory cells are produced which respond rapidly when infection occurs. In terms of passive artificial immunity, antibodies are formed in one individual. It's extracted and given to another individual. So the natural immune response is not stimulated. The protection is short-lived, only for about 2 weeks. It gives immediate protection in the case of an emergency, but no memory cells are produced. All right, so this is it as it concerns immunity, guys. So we have one more topic, which is to look at health and disease. So I'm going to give you your final break, and then we look at that topic.